Welcome back once again to my educational channel on biology. We are continuing with our REV UP series for SPM, which is a series of revision videos on different chapters in Form 4 and Form 5 biology syllabus. So we'll continue with chapter 4, subtopic 4.5 on nucleic acids, which are DNA and RNA. Now this topic tends to be difficult to understand and master for some students. But do not worry, as here I've included a mind map to summarize our concepts and organize all our ideas and what we have studied. So this should be able to help you well. Okay, and apart from that, we will also discuss some very good questions from the trial papers on nucleic acids. It will train you to answer these questions well in the SPM and to increase your performance or improve your performance in the SPM. Right? So let's begin. So firstly, we're going to look at the concept map for Form 4 chapter subtopic 4.5, okay, on nucleic acids, that is DNA and RNA. Right, so let's look at the mind map. So mind map is a picture that summarizes all the important points, okay, that are divided into various subtopics, right? And it will give you a good idea of all that you have studied for this subtopic, okay, and help you to memorize it. So the first section is on the elements. What are the elements of nucleic acids? So nucleic acids such as DNA and RNA are made up of the elements C, H, O, and P, which are carbon, hydrogen, oxygen, nitrogen, and phosphorus, right? Next, we go on to the basic unit of nucleic acids in the cell. So the basic unit is called the nucleotide. Okay, it is a monomer, but many of these monomers will be joined together to form a polymer, which is the DNA and RNA. So both DNA and RNA are made up of the basic units called nucleotides, which are joined together. Now let's look at the structure of one nucleotide as shown in the diagram here. So the nucleotide consists of three parts: the phosphate group, pentose sugar, that means 5-sided sugar. Okay, now there are 5 sides here, and one example is the 5-carbon sugar called deoxyribose. Okay, then we have the nitrogenous base. Now let's look at each part in more detail. The phosphate group, which consists of the elements phosphorus and oxygen, right, is the first part. Okay, it has the phosphate in it. And then we have the second part, which is the pentose sugar or 5-carbon sugar. Now there are two types of pentose sugars, which are the ribose sugar found in RNA. That's why it's called ribonucleic acid. The ribo comes from the word ribose. Okay, ribose sugar or ribonucleic acid. And for DNA, we have the D here stands for deoxyribose sugar. Okay, so RNA contains the ribose sugar and DNA contains the deoxyribose sugar. Okay, that's why DNA is called deoxyribonucleic acid. Okay, the deoxy comes from the word deoxyribose. Right, then as for the nitrogenous base, the third component, there are actually 5 types of bases that exist: adenine A, guanine G, cytosine C, thymine T, and uracil U. Okay, right. Now, all these 3 parts are joined together by condensation to form the nucleotide, as we can see here. Okay, now you can see the phosphate group is joined to the pentose sugar, which is joined to the nitrogenous base. The pentose sugar is in the center. So you have to know how to draw this structure, okay, which is a nucleotide. But the DNA molecule is not just made up of one nucleotide, it is made up of many nucleotides joined together. Similarly for the RNA molecule. So we have discussed the basic unit. Let's go on to the third aspect, which is the types of nucleic acid. Right? The types of nucleic acid include ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. Okay? So we have to know the characteristics of RNA and DNA. Now, firstly, DNA. This can come up for essay question. You can be asked to describe or explain the structure of DNA as shown in this diagram here. Okay? So the basic unit for DNA is the nucleotide. Now, if you want to discuss further about the nucleotide, you have to refer back to this part here. Okay? So refer to nucleotide. That means you have to mention the nucleotide. Each nucleotide consists of 3 parts, which are phosphate group, pentose sugar, and nitrogenous base. Alright? And then many nucleotides join together to form a polynucleotide chain or strand. Now you can see here, this is one nucleotide. It's joined to the next one below and the next one below here. So this whole chain is called a polynucleotide chain. Alright? The phosphate group of one nucleotide is joined to the pentose sugar of the other nucleotide. Okay, now this phosphate group of this middle nucleotide is joined to the pentose sugar of the nucleotide above it. Okay, so the pink and yellow structures here, the phosphate group and the sugars, form the backbone, right, of the molecule. Now, but DNA has 2 polynucleotide chains. Okay, now this one here and this one here, right, which are intertwined. Intertwined means they are twisted like this to form a double helix structure. So this is an important keyword. 2 polynucleotide chains, okay, one here and the other one here, that are intertwined to form a double helix structure. Okay, so I say this word, double helix structure. So apart from that, where are the nitrogenous bases, which are adenine, thymine, guanine, and cytosine, ATGC? They are right in the center between the 2 polynucleotide backbone here. Okay, so this is one polynucleotide chain, right? So it has its bases here, and this is the other polynucleotide chain. Its bases are also in the center. So the bases from the 2 polynucleotide chains are joined together. They pair up in the center, bound by the hydrogen bonds. Okay, the weak hydrogen bonds. All right. Now take note that the 2 strands of polynucleotides are parallel to anti-parallel, not parallel, anti-parallel to each other. What do you mean by anti-parallel? You can see that the pentose sugar of one polynucleotide strand will face upwards, but the other one will face downwards. See, the pointed end faces downward. Okay. Now this is important to note because sometimes you will be asked to draw. You may be asked to draw maybe one part of the polynucleotide chain. You may be given one part and be asked to draw the other section. Okay, as you will see in the exercise later on in the revision questions. So adenine, for DNA, adenine pairs with thymine and guanine pairs with cytosine. Okay, so adenine cannot pair with cytosine. Adenine only pairs with thymine and guanine only pairs with cytosine. Then the backbone is made up of the sugar and phosphate groups. Okay, now you can see here, this is the backbone, sugar and phosphate groups. Okay, phosphate, sugar, and so forth. Right, so this diagram is quite important for you to memorize how to draw. Next, let's go on to ribonucleic acid. Ribonucleic acid is also made up of the basic unit called nucleotide, and it consists of only one polynucleotide strand. Okay, as you can see here, compared to DNA, there are 2 polynucleotide strands here. Okay, now the nitrogenous bases are adenine, uracil, guanine, cytosine. So notice that there's no thymine here. So in RNA, the thymine is not there. It's replaced by uracil. We can say that thymine in DNA has been replaced by uracil in RNA. You don't get a T, you get a U instead, all right, as one of the bases. Adenine pairs with uracil, A to U, and guanine pairs with cytosine, G to C. Okay, right, discussing the structure of the RNA, also mentioned that the basic unit is the nucleotide, which is made up of 3 parts, phosphate group, pentose sugar, and nitrogenous base. Right, so more facts about ribonucleic acid first. Now, ribonucleic acid consists of 3 types. There are 3 types of ribonucleic acid, or RNA, that are used in protein synthesis. The first one is called messenger RNA, or mRNA. M comes from the word messenger. Messenger RNA copies the code, the genetic code from DNA in the nucleus, right? Then ribosomal RNA is found on the ribosome. It's called RNA also, rRNA, and it is found in the ribosome itself. Then you have transfer RNA. Transfer RNA is a type of RNA molecule that brings the right amino acid to be joined to form the polypeptide chain. It will bring the amino acid to the ribosome. Okay, that's why it's called transfer. Transfer the amino acid from the cytoplasm to the ribosome. Now, we do not need to know specifically how they carry out their their functions, but you must know that there are 3 types of RNA. All right? Right, so next, what about the function? Function of RNA is it uses the genetic information that it has copied from the DNA in the nucleus to synthesize protein. Its main job is to help in the synthesis of protein on the ribosome, which is an organelle that's a bit rounded in shape. So RNA helps in the process of synthesis of protein, as we'll discuss in the next slide. Now, for DNA, its main function is to store genetic information in the nucleus. Okay, so it doesn't come out of the nucleus. It stays forever in the nucleus and has a store of the genetic information that's very important to keep well inside the nucleus. Right, so next, let's go on to the next aspect, the importance of the nucleic acids. Now, let's talk about DNA first. DNA is the carrier of hereditary information. It determines the characteristics in the organism, so it's called a determinant of characteristics in the organism. And also, the word hereditary information means that DNA has genetic information for the synthesis of proteins that can be passed down from parent to the child. Okay, that's why it's called hereditary. Boleh diwarisi. Boleh diwarisi. Can be inherited by the child from the parent. So the DNA can be passed down, right? A copy of the DNA can be passed down from parent to child. Now, the third point is that it also stores genetic information about protein synthesis. Okay, it stores genetic information about protein synthesis as genetic codes. So the code is carried in the sequence of bases. So the code means there is a way in which the information is stored and is in the form of a code. And this code is found in the sequence of bases ATGC, as we'll see afterwards. Okay, so the code will tell the cell what type of amino acid to use to join together to make the protein. Okay, so we'll discuss that later. The codes are used for protein synthesis in the cell. Okay, so DNA stores the genetic information or code for protein synthesis in the cell. Whereas RNA uses this genetic information from DNA. It copies the genetic information from DNA in the nucleus and brings it out to the ribosome to help in the synthesis of polypeptides. And then when these polypeptides are formed, there may be some folding and some more change in structure, then proteins will be produced. Now, the structure of nucleic acids in general is that they are made up of 1 or 2 polymer chains. That means 1 or 2 polynucleotide chains. Okay, polynucleotide chains that comprises the nucleotide monomers that comprise this nucleotide. Okay, so for RNA, it is made up of 1 polynucleotide chain, and for DNA, it's made up of 2 polynucleotide chain. But inside the chain, the basic unit is the nucleotide monomer, which is this one, the nucleotide. So I hope you have a clear picture now of what we have discussed here. This is the first part of our summary, and you will find this useful. Do give me some feedback on this so that I can continue to do more of these mind maps for you in the future. All right, so give me some feedback whether you find this very effective for you to use. Now the second part, okay, this is the last part that we are going to go through. Nucleic acids, how are they used in the synthesis of proteins first? Okay, now the genetic code is a series of 3 bases or codons, such as adenine, uracil, guanine. So it's AUG. GCA is guanine, cytosine, adenine, or CCG. Okay, cytosine, guanine. So in the polynucleotide, all right, the genetic code is in the form of the sequence or the series of bases in the center part of the DNA molecule. All right, now you can see here, if this is the DNA molecule, let's look at the diagram on your right here, right? So in the center part here, you have the base pairs. So you look at one side, all right, the sequence of bases on one polynucleotide strand, it may be AUG, all right, and then further down it may be GCA, you see, and then maybe something else. So these are all the series of 3 bases, okay, 3 bases, 3 bases, 3 bases, and so forth. Now, each group of 3 bases in mRNA is a codon that specifies a specific amino acid. How did the mRNA have bases? Actually, the mRNA copy the bases from the DNA in the nucleus. The DNA is like a storehouse with all the codes of 3 bases or codons. Then the mRNA will copy the code from the DNA. Okay, so each group of 3 bases is a codon. Codon means a code that specifies a specific amino acid. It stands for a specific amino acid. For example, we said AUG. Let's say AUG on the one side of the, or one nucleotide. What does it stand for? It stands for methionine, which is an amino acid that needs to be joined to form the polypeptide chain. Okay, so it's like an ingredient that's used to make a cake. All right, so this one is the item that's needed to make the polypeptide chain. Okay, so the cell will need to bring this methionine from the cytoplasm to join to the growing polypeptide chain, okay, on the ribosome. Now, so sequence of nucleotide bases on DNA is copied onto mRNA, and then it will determine the sequence of amino acids in the polypeptide chain and protein that is formed at the ribosome there, in the cytoplasm of the cell. Okay, now let's look at the process. There are 2 parts, okay, 2 basic parts to this process. The first part in the synthesis of proteins is that in the nucleus, the 3 base sequences in DNA is copied or transcribed. Okay, the right word is transcribed. That means it's copied into the mRNA to form the mRNA codons or codes. Okay, now, okay, let's say you look at this diagram on the right. Now, so in the nucleus here, you have all the chromosomes. And on the chromosomes, if you were to unravel the chromosomes, you can see that they are made up of DNA, right? So first of all, this double helix structure of DNA has to unwind. Okay, so it open up. So it will reveal its code, okay, on one polynucleotide strand. So let's say the code is TACCGT. That means thymine, adenine, cytosine, cytosine, guanine, thymine, right? So the first 3 amino acids here are TAC, right? Right? So the mRNA is formed by copying the code on DNA, but it doesn't copy exactly the same bases. When it's produced, it will have the complementary bases. If on the DNA, the first base is T, now T only binds with A, right? Thymine only binds with adenine. So on the mRNA molecule, the complementary base will be A instead, as it copies the code. Okay, then A will bind with U. In mRNA, there's no T, there's only U, uracil. Okay, so when the code is formed on mRNA, it will be uracil here. Okay, and when the code is copied, and then C, look at the complementary base is G, right? And then we have the C here again, so complementary base is G on mRNA, right? And then and so forth, right? So this is the process of transcription in the nucleus, all right, to form the codons on mRNA. So each codon is made of 3 bases. So this mRNA will leave the nucleus and go to the ribosome, and it will sit on the ribosome, and then the code is translated. So the second part is translation. On the ribosome, the mRNA that has come out from the nucleus into the cytoplasm, the mRNA codons are read and translated into a sequence of amino acids to form a polypeptide chain and then the protein molecule. Okay, so as the ribosome, as the mRNA is on the ribosome, the code is read. So AUG actually stands for the amino acid methionine. Okay, and then GCA stands for the amino acid alanine. So these 2 amino acids will be joined together. Okay, and then as the code is read further, more amino acids are joined together. So this process is called translation. That means the code is read and translated to form the to know what is the amino acid that needs to be brought to join, okay, to the growing polypeptide chain. Finally, a long chain of amino acids will be joined together, and this will form the polypeptide chain. Then with all the folding and other bonds being formed, finally the protein molecule is formed. Okay, right. So remember, the process of protein synthesis involves transcription and translation. Okay, transcription in the nucleus, translation on the ribosome. Now, finally, formation of chromosomes. So here we are going to discuss how the chromosomes are formed. What is the structure of the chromosomes? Now, chromosomes actually form from two components, DNA and histones. Histones are the proteins, okay, that do not carry genetic information. Only the DNA contains the genetic information. So what are the histones used for? Okay, the DNA is wound around the histones. So histones act like a spool. Now when you wind your thread round and round in order to store it and keep it prevent it from being entangled. Okay, so when DNA winds around the histones, they form the nucleosomes. Okay, now these are the nucleosomes as shown here, and you can also see it here. So the blue strands are the DNA, is a single DNA molecule that's very long, so it's wound around the histones here. So the histones are made up of a few parts here, right? And then histones have to be intertwined. That means they're twisted together and sort of grouped together like this, right? To finally form the chromosome. Now, this is all to make sure that the DNA molecule that's very long is not entangled up or damaged in the nucleus or in the cell. Okay, so it's carefully wound up so that it is well organized. Okay, during the process of cell division, it's also very carefully well organized and prevented from being damaged. Okay, that's all for this short revision. It's only taken us a while, about 8 minutes or so. And if you have any questions related to this, you can ask. If not, you can copy down this mind map, which is very easy to understand and to revise, very quick for you to revise. Okay, so these are all the important points in the notes. Now let's go on to the questions. Our first question. The diagram 2.1 shows the structure of a type of nucleic acid. Name the parts labeled P and Q. So you can try this question first, and then we can look at the answers afterwards. Okay, you can pause the video and try the question first. Right, let's discuss the answers. Now, name parts labeled P and Q. What is P? Okay, P are the hydrogen bonds, right? So P is a hydrogen bond here, a weak hydrogen bond that binds the two bases together. Okay, such as adenine and thymine, right? So this bond between them is called the hydrogen bond. And how about Q? Q is the 5-sided sugar, which is called the deoxyribose sugar in the case of DNA. Okay, this molecule is a DNA molecule because it has 2 polypeptide chains, one on each side, right? So that means the sugar is not ribose sugar, it's deoxyribose sugar, right? And the general term is pentose sugar. So I would advise you to write the specific name first, and if you are not sure whether to include the general name, just write it in brackets. Okay, so both may be accepted, but the more specific name is deoxyribose sugar. Now let's look at the second part. State the function of the nucleic acid in diagram 2.1 in an organism. So what's the function of the DNA molecule? 1 mark only, so just give one function. Let's look at what the answer is based on the textbook, right? So most answers will be based on the textbook. So make sure that our notes are based on the textbook also. Okay, so these are the answers based on the textbook. Firstly, it is the carrier of hereditary information. That means information, genetic information that can be passed down from parent to child. Secondly, it determines the characteristics in an organism, or you can say it's a determinant of characteristics in organisms. For example, blood type in humans. Now, thirdly, it stores genetic information for protein synthesis in the cell. Okay, so the genetic information here is about the synthesis of proteins, different types of proteins in the cell. Okay, so any 1 point, 1 mark. Next, state one characteristic of the nucleic acid in diagram 2.1. What's the characteristic? Okay, first was the function, now characteristic, right? So you can talk something about its structure, right? 1 mark only. So answer is consists of 2 chains or strands of polynucleotides. Okay, one on each side here. One here, one side here, and the other side here. Now next, these 2 chains intertwine, that means they are twisted together to form a double helix, which is shaped like a spring, right? So the word double here means got 2 strands of polynucleotides. Another point you can write is that it has 4 different types of nitrogenous bases, and then just take the names: adenine, thymine, cytosine, and guanine. No uracil, okay, because this is DNA. DNA doesn't contain uracil. Okay, so any 1 point you get 1 mark. All right. Now, if you're not sure, just write 2 points down. All right. But the more important point should be at the top. Should be the first point. Next question. The diagram here shows the processes that occur during protein synthesis. So explain the effect on protein synthesis if the transcription process fails to occur. Okay, that's why we need to know the meaning of the names transcription and translation as shown here. So what happens if the transcription process does not occur at the top here? So we know transcription means the copying of the code from DNA to RNA, right? So this is a hot question. What happens if there is no transcription? So all the rest of the processes will not occur, right? Now, firstly, you can think about the answer first and write it down first. Pause the video. Okay, let's look at the explanation. Now, it's 2 marks, 2 to 3 marks. So the first point is that the mRNA cannot be formed because to form the mRNA, we have to copy the code from DNA through transcription. So in this case, the mRNA cannot be formed because there's no transcription process for the mRNA to be formed by copying the code on DNA. Okay, so no codon is formed. You don't get all these codons like AUG, CGA, and so forth. So the translation process, the next process will not occur at the ribosome. Okay, that means the code cannot be read and translated to know what is the specific amino acid to join together. The specific amino acids to join together. That means the protein cannot be synthesized. Okay, there's no gathering of the amino acids and the joining of the amino acids at all because there's no code. The cell, the ribosome doesn't know what's the code, right? So there's no mRNA, there's no code. So protein is not synthesized and there's no production of polypeptides. Okay, so any 2 points will be 2 marks. So good question. Please take note of this question. Going on to question 2, diagram 1.1 shows the structure of a type of nucleic acid. So name the type of nucleic acid. And the second question is, the nucleotide is the basic unit for nucleic acids, so draw and label the structure of the nucleotide. Okay, you can pause the video and try the question first. Right, let's look at the answer. Name the type of nucleic acid. So you can see that this is a double helix structure and there are 2 polynucleotide strands that are twisted, intertwined or twisted together. Okay, so the type of nucleic acid is deoxyribonucleic acid. In brackets, the short form is DNA. So students ask whether we can just write DNA. No, you have to spell out the full name. As I said, if somebody asks you what's your name, you say DNA, nobody's going to know what it actually stands for right now. So it must be the full name, deoxyribonucleic acid. If not, you can't get your 1 mark. So the DNA, the name DNA can be written in brackets, right? So remember, deoxyribonucleic acid. Now let's go on to the second question. The nucleotide is the basic unit for nucleic acids. Draw and label the structure of the nucleotide. Okay, remember, nucleotide, basic unit for nucleic acids. All right, so have you drawn your diagram? Let's look at the structure. Okay, so here we can see that there are 3 components in the structure of the nucleotide. Okay, this is just one basic unit, one nucleotide. So we draw a circle to represent the phosphate group, 5-sided structure to represent the deoxyribose sugar. If you write pentose sugar, they also accept, but the more specific name is deoxyribose sugar. And then you have the nitrogenous base. Okay, the shape, it can be just a rectangle also. Okay, here I show the shape with a special edge here, okay, to match another shape on the other side. So correct diagram, 1 mark. Okay, and then that means all these parts are joined together in the right sequence, the right phosphate group first in the right position. Then sugar, the sugar is in the center, and then the base is at one end. 3 correct labels, 1 mark. So altogether 2 marks. Okay. Now, if you don't label the diagram, is it okay? No, if you don't label the diagram at all, or your label is wrong, any of the labels are wrong, you will lose 1 mark. Okay. So for this question, again, pause the video and try the 3 questions here before we discuss the answers. Right, let's look at the answers. Now, explain the role of nucleic acids to organisms. So add the word this, explain the role of this nucleic acid to organisms, which is the DNA, as was mentioned in the first part of the question. Okay, so the role of DNA. So it is the carrier of hereditary information. It determines the characteristics in organisms, for example, blood type in humans, and it stores genetic information for protein synthesis in the cell. These are all the functions of DNA, as mentioned in the earlier question. Now, if it is functions or just roles of nucleic acids in general, then we can also state for RNA, the role of RNA, especially mRNA, is that it is directly involved in protein synthesis at the ribosomes. Okay, involved in protein synthesis at the ribosomes. That is for mRNA's function. Now let's go on to question C. Diagram 1.2 shows the sequence of nitrogenous bases in a polynucleotide chain. Question, state the match for the nitrogenous base sequence of the polynucleotide after the process of transcription. So we know that in the process of transcription, the genetic code on DNA is copied onto mRNA. Okay, so this is the code from DNA. It has thymine, right? And let's say it's copied onto mRNA. Now, the code is not exactly the same like DNA. It's actually the complementary bases that are involved, all right? The matching bases. So, in place of A, the base in mRNA will be uracil, U, okay? Because mRNA doesn't have thymine. So, U pairs with A. Now, T here, so the base on mRNA will be the complementary base, which is A, adenine, okay? So, UA, and then complementary base for G is C, cytosine, right? Now, if it's cytosine, then the complementary base is G, guanine. And then these two are thymine and thymine, so the complementary bases will be adenine, adenine. So our answer will be UAC, UAC, and then a blank space here, and then GAA, okay? These are all the complementary bases of the bases here, right? Now, don't use thymine, don't put thymine in place of uracil because mRNA doesn't have thymine. Okay, let's go on to the last question here. The diagram below shows a nucleotide. Complete the diagram by drawing another nucleotide on the right-hand side here to show a part of a DNA molecule. Okay, so assume that this nucleotide is from one polynucleotide strand, and then opposite there's another, on this side there's another polynucleotide strand with the nucleotides involved. So how would you draw the nucleotide here? Is it exactly the same like this? No, it's different. Let's look at the answer. The nucleotide will be facing with the pointed end facing the opposite way. So although it seems parallel, but it's actually called anti-parallel. Okay, that means the orientation of the deoxyribose sugar is different for this nucleotide here and this nucleotide here. Now you can also label the parts. Original picture. Okay, so notice that the deoxyribose sugar for this nucleotide is facing in the opposite direction, or in another direction compared to the first one here. Question 3, last question. So diagram 1.1 shows an animal cell, okay, here. And you are asked to name the structure X here, okay? Okay, you can read through this question first and try it. All right, let's look at the answers. Now, name structure X. What is structure X? It's an X-shaped structure, right? It's called the chromosome, right? Now, next, structure X is formed from DNA polynucleotide chains that are intertwined with proteins called histones. Complete the nucleotide structure in the box below. Again, it's the nucleotide structure, right? So what's your answer? Okay, try and draw and complete this diagram to show the nucleotide structure. And then also answer question 2. Question 2 here, okay? Right, let's look at the answer. So you have to add in the nitrogenous base and the phosphate group on both sides, on the two sides. So you look like this. Right, now so draw the phosphate group, label it on one side, and draw the nitrogenous base on the other side of the sugar. Right, label it nitrogenous base. So since it's 2 marks, it's 1 mark for each component. Right, phosphate group, 1 mark, and nitrogenous base, 1 mark. All right, next, there are 2 types of nucleic acids that, and they are DNA and RNA. Compare these 2 structures, compare these 2 molecules, right? So here the table has been drawn for you. In the upper part here is actually for you to write the similarity. Now you want to compare, you must have similarity and differences. Here is altogether 3 marks, right? So at least have 1 similarity and 1 difference. But since the similarities are more, maybe they expect 2 differences here. Now I can see 2 differences, 2 boxes here for 2 differences, and then 1 similarity. Okay, so since the box is given, the table is given, you can just fill it out. But if the table is not given, it's just paragraph form, then you have to write the compare in a paragraph form. Okay, but here the table is given. All right, what is the similarity first of all? Okay, let's have a look. So try the question first. Similarity, there are 2 here. Firstly, both RNA and DNA and RNA contain the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus, which is CHOMP, C-H-O-M-P, CHOMP. Okay. Next, the basic unit for both DNA and RNA is the nucleotide, which consists of 3 parts, which are the phosphate group, pentose sugar, and nitrogenous base. Okay. Now next, let's go on to the differences. So you need 1 similarity and 2 differences for this answer. So there are many differences. Let's just go through, and this can also come out in the essay question, this whole thing on the similarity and differences. Okay, so you can copy this down into your notebook as part of the revision. Okay, the differences and similarities between DNA and RNA. So number 1, DNA strand is longer and RNA strand is shorter. Number 2, DNA consists of 2 polynucleotide strands, but RNA consists of only 1 polynucleotide strand. Number 3, DNA has a pentose sugar, which is deoxyribose, but RNA has a pentose sugar, which is ribose. Okay, its type of pentose sugar is ribose. Number 4, the nitrogenous bases in DNA are adenine, guanine, cytosine, and thymine, but the nitrogenous bases in RNA are adenine, guanine, cytosine, and uracil. Number 5, this is on the location. DNA is located in the nucleus and mitochondria of the cell, but mainly in the nucleus. Whereas RNA is located in the nucleus, but it will come out of the nucleus and it will go into the cytoplasm, okay, to carry out its function. So mainly in the cytoplasm, right? Now next, DNA stores genetic information which can be inherited by the child, but RNA is not for storing genetic information permanently or for a long period of time. It is just to copy the genetic information, and then it is used in protein synthesis, where the code on it will be translated, all right, to know what's the sequence of amino acids needed in protein synthesis. So it's directly involved in protein synthesis at the ribosome. Right, that's all for this lesson. Thanks for viewing. And here we can see the 3 different containers where the DNA has been extracted from different fruits. The DNA is the white stuff shown here. So this was done in a school project, all right, which needs teacher's supervision. And you can see the DNA, which is a macromolecule. It has come out in the form, sort of solidified in this form, the whitish substance here, all right. And these are the DNA from various fruits, right? Like strawberry, orange, and kiwi. So thanks for viewing, and do share, like, and subscribe. And all the best to you till we meet again. Goodbye for now.